Morning, viewer. Welcome to our Glory Globe TV as a breaking news. Viewers, researchers at Yale School of Medicine have discovered unexpected insights into how the eye handles visual information. When we view a scene, the visual system quickly separates different features, including color, contrast, and motion too, and also processes them independently. And this process is known as parallel visual processing helps the brain rapidly interpret the world about us. Scientists have long believed that this separation begins in the retina and remains intact as information moves through the visual system. However, a new study published in Neuron suggests the pathways are more interconnected than previously recognized. According to the researchers, this integration may improve the ability to detect weak visual signals such as those encountered in low light. We found that while different channels can deliver their own features, they are also interconnected by underlying electrical circuitry, says Yao Zihui, PhD, a postdoctoral fellow in the Department of Ophthalmology and Visual Science at YSM and the study's first author. Vision. Viewers, vision starts with rods and cones, specialized retinal cells that detect light and pass information to neurons known as bipolar cells. So within these cells, visual information related to factors such as brightness, color, shape, and contrast is divided into more than a dozen parallel pathways. So when the researchers examined the synapses of bipolar cells, where cells communicate with one another, they found evidence that these pathways are not entirely separate. Neurons communicate through two types of synapses, chemical and electrical. Chemical synapses rely on neurotransmitters that carry signals between cells. Electrical synapses, also called gap junctions, allow signals to pass directly through electrical currents. So bipolar cells are generally thought to communicate many through chemical synapses. The researchers discovered that electrical synapses connected many of the bipolar cell pathways that had previously been considered separate in both mouse and human retinas. When they stimulated a single bipolar cell, the resulting neurotransmitter releases was not confined to that cell's pathways. Instead, signaling spread across a wider network, creating diffuse, cloud-like activity patterns that enabled extensive communications between different bipolar cell types. So when we stimulated one bipolar cell, many bipolar cells released neurotransmitters, says Z. Jimmy Zhao, PhD, Marvin L. Sears, Professor of Ophthalmology and Visual Science and Principal Investigator, behind this exclusive report.